Okay, everyone. Now we are going to continue our discussion on Chapter 6, which is Stereochemistry in Chem 201. And so I'm going to start by sharing my iPad. When we left off, we had just finished a pretty intensive discussion on enantiomers and also how to differentiate between those two enantiomers using the R and the S designation for the configuration of the chiral center. What we're going to do now is what happens when I have two or more stereocenters within the same molecule. As you can imagine, once I start introducing more stereocenters, the picture gets a lot more complex. In fact, if a molecule has N stereoisomers, the maximum number of possible stereoisomers, so these again are going to be same connectivity, different orientations in space, is 2 to the nth. So this can get really large really quickly. Remember, if my molecule only has one stereocenter, I get two stereoisomers, which is one pair of enantiomers. Here's where we're going to focus next. If I have two stereocenters, I can get up to 4 stereoisomers. I actually get 2 pairs of enantiomers that are possible. In this course, since it's not the organic chemistry 1 and 2 sequence for science majors, we pretty much stop here in terms of discussing this type of concept, but just recognize I could have as many stereocenters as I have carbons in some regards. So if I have a protein and I have maybe 450 amino acids, I could have 450 or 450 chiral centers if I consider that every amino acid except for glycine has a chiral center. So again, this can be extremely large numbers that are possible. So for example, if I have 3, I can get up to 8 stereoisomers or 4 pairs of enantiomers. So let's look at this more carefully. This is 2, 3, 4. Remember, I have 1, 2, 3, 4. This is actually an aldehyde. I have 2 stereocenters, one on carbon number 2, one on carbon number 3. So therefore, I can get up to 4 stereoisomers that are possible. So let's look at what they could be. I'm going to use my hash wedge and my bolded wedge in order to indicate stereochemistry. Remember, the bold is coming out at me and the hash is going behind the plane of the paper. So here's one that I can draw where I have the aldehyde on the top and the alcohol on the bottom. This is where I have my stereochemistry centers. What if I have both of these hydroxy groups bolded pointing out at me? Now, the mirror image to that is going to have bolded hydroxys, but coming off to the left of those carbons. So this is one pair of enantiomers. But let's say I keep this the same, but I change this one. So now I have my bolded OH on opposite sides of this sort of axis down the middle of the compound. Well, this stereocenter is going to be the same. This stereocenter is going to be different. And then to compound that, I can actually draw the mirror image of this, where now I have bolded right and bolded left in terms of my hydroxides. This is my second pair of enantiomers. The thing is, this and this are called diastereomers. This and this are also diastereomers. I could also say that this and this are diastereomeric, and this are diastereomeric. So I have two pairs of enantiomers, but each enantiomer compared to the other two stereoisomers of the second enantiomeric pair are going to be diastereomeric in relationship. So what does that mean? That means that I have to really look at this and focus on the fact that when I have diastereomers, they're not mirror images. If I look at this guy and I look at this guy, I have a bold on my number 2 and 3 carbons. The number 2 stereocenter is the same, but the number 3 stereocenter is different in this compound. That makes it not the mirror image. I have the mirror image is this going bolded to the right, this bolded to the right, this folded to the left, this folded to the left. See how this is the mirror image. But this is the same direction. This bottom part is the mirror image of that, but this top part is going to be the same. So basically, enantiomers are going to have opposite configurations. So if one is R and the bottom is S, the enantiomer will be SR. It'll have opposite configuration. So this and this will have opposite configurations. This and this will have opposite configurations. The two together having opposite configurations make that enantiomers. This configuration is the same as this one, but this is not. So when one is the same and one is different, that is where I'm going to have diastereomers. Okay, here's an example before we get into that topic. Mark all the stereocenters in each molecule and tell how many stereocenters are possible. Well, this one only has 1, so 2 to the first, I'll have 2. This one only has 1. This has 2 methyl groups, so that's not a stereocenter. So this has 2 to the 1 is equal to 2. This is a stereocenter. This is a stereocenter. This has 2 to the second, or equal to 4. This has 1, so it's 2. This has 1 and another 1, so I have 4. And this also has 2 stereocenters, so I have 4 possible stereoisomers. So again, you have to be good at recognizing which carbon has 4 different attachments. And you're going to have to start thinking about how I can have more than one of those stereocenters like I do in many of these. Okay. So let's take an example problem and let's find and draw all possible stereoisomers for a compound that has 2 chiral centers. The first thing I want to do is draw one stereoisomer. And I'm going to arbitrarily arrange the substituents around that stereogenic center. Then, when I get that, I'm going to draw its mirror image. So here, I have 2,3-dibromopentene. So let's look at that. 1, 2, 3, 4, 5. I have 2,3-dibromopentene. So this is a methyl. This also has a hydrogen. This is a stereocenter. This has a hydrogen. And then I have this ethyl group. This is a stereocenter. So I'm just going to pick one, right? I'm going to say, all right, well, I'll just put the bromine in my dashed wedges, or my bolded wedges, and put the bromines on the same side. Well, then I'm going to draw the mirror plane. Remember, I have to add everything up. So this, or I have to reflect everything. So this hydrogen is coming backwards. This bromine is coming towards me. This part, this hydrogen has to go back. This bromine has to go forward. This and this are enantiomers because they are mirror images of one another. Now, I'm going to check to make sure that I have enantiomers. I want to make sure that those are not superimposable mirror images, right? So I'm going to take one and rotate like we did before. And what I'm going to find out is there is no way that A and B are going to be superimposable on one another. Therefore, A and B are non-superimposable mirror images, which makes them enantiomers. So I have one set of enantiomers, and A and B are 2 of the 4 possible stereoisomers. So how do I get the other 2? I'm going to switch the positions of one of the hydrogen and bromine on one of the stereogenic centers of either A or B. So let's work with A, where I have these bromines bolded out. I'm going to switch this center. So now I put the bromine hashed backwards and the hydrogen bolded forwards. So this is not the same as A, and it's not the mirror image because the only thing I changed was this. This stays the same. Now I could have arbitrarily changed that one and left the other one stay the same. I can just pick one. Then I'm going to draw the mirror image. The mirror image of this is going to have, right, this hydrogen reflects this, the bromine coming out reflects this, these 2 reflect, these 2 reflect. I have the bromine coming back at the end, and then that last one has the hydrogen coming out. So C and D are mirror images. These are the second pair of enantiomers. Now, if I go A to C, or A to D, or B to C, or B to D, I have stereoisomers that are not mirror images. So those groupings are diastereomers. So again, I could take A related to C, or A related to D. They are diastereomers of one another. A and C and A and D are diastereomers. I could also do B and C or B and D. So B and C and B and D are also diastereomers. So A and B is one set of enantiomers. C and D are the second set of enantiomers. Remember, I arbitrarily draw the mirror image. I change one of the two chiral centers, and then I draw that mirror image. That gives me A and B and C and D. Those are those two pairs of enantiomers. A and C, A and D, B and C, B and D, those are my diastereomeric pairs. I have one chiral center that is the same and one that is different. If I look between A and B, this chiral center is different. This chiral center is the same. If I look between A and D, this chiral center is the same, and this other chiral center is different. That's how I get those different relationships. So this is sort of the relationship. I have my two sets of enantiomers, and then any other relationship is going to give me diastereomers. So this is a pretty good diagram to look at. One enantiomer, one chiral center stays the same, the other is different. Now I could assign configurations. And let's say I look at A, B, C, and D. Let me use a different color. If I look at those two chiral centers that are mirror images, so A and B, and then I look at C and D. I'm not going to draw specifics here. I'm just going to give you a generality. If this would be RR, this configuration would have to be SS. If this is RS, this configuration has to be SR. So when I have enantiomers, the two chiral centers in one molecule is going to be the opposite configuration of the two chiral centers in the enantiomer. In diastereomers, so look at that, what I mean by that is I have RR on A, and let's say I have SS on B. This is hypothetical. I didn't actually assign the configurations to my actual molecule. In the other set of enantiomers, let's say I have R, that the top one that stays the same, and now S is going to change. The enantiomer, the top one would be opposite, or S, and the bottom would be opposite, or R. Anytime I have one the same and one different, like if I have R across the board, but then I notice this is R and this is S, this would be diastereomeric. I always start with the two enantiomers, and then the relationship between one enantiomeric pair and the second enantiomeric pair will be diastereomers. So this 2N rule applies equally well when I have 3 or more stereocenters. Here I have 3 stereocenters. I could get a potential of 8. Menthol is one of those 8, and there's a couple different ways. We learned about how I could take that and draw the chair configuration. So these are actually equivalent structures, I just have one in the chair, and this is going to be the more stable chair because notice that all of the three groups are equatorial. This is cholesterol. I have 1, 2, 3, 4, 5, 6, 7, 8 stereocenters in cholesterol. So I could have 256 possible combinations. Only one is found in human metabolism. This is something that's really important. This is a really important feature of bioorganic or biochemistry. Nature is extremely stereospecific. Even though I could have a potential of 256 stereoisomers, only one is used in the biology of the human, right? And that's what we're going to see in proteins and in any other chiral system that we see in biology. Now, just to be contrary, there is something called a meso compound. And this is an achiral compound that has 2 or more stereocenters, but the mirror image is actually superimposable, which means that instead of being stereoisomers, they are actually the same compound. So let's look at this. Here's my original pair of enantiomers for this compound. But notice on top and on bottom, I have the same groups. I have a carboxylic acid here, a carboxylic acid. Well, I draw the mirror image. I have bolded right, bolded left. The OH here is bolded left, bolded right. When I keep one the same and change the other, so I just changed this one to get to this structure, I actually have a plane of symmetry within the molecule. Even though I can draw the mirror image, because this has an internal plane of symmetry, these are actually the same compound. So it's achiral, and it's called meso. So this example only has 3 stereoisomers because this fourth is not a separate compound. It's identical to the third one. So really, I only have 3 stereoisomers because these two, that is meso, which means it's the equivalent. To find meso compounds, I'm going to look for internal planes of symmetry. An imaginary plane that passes through an object and divides it such that 1/2 is the mirror image is what I'm looking for. That's a plane of symmetry. So for example, if I have a cylinder, and I put this pink plane in there, 1/2 reflects the other. When I have cubes, I have, in this example, 3 different planes of symmetry. The yellow plane of symmetry, the pink plane of symmetry, and the blue plane of symmetry. It's the same thing when I have a molecule. When I have meso, I have an internal plane of symmetry where this is reflecting that. Sometimes I cut through atoms in order to get that plane of symmetry. So these are in sort of the plane, and this is reflecting this, which gives this the internal plane of symmetry. When we look at cyclohexane molecules, we're going to see that we can have many examples. If I have, and what I'm looking for is symmetry. In this example, which I have cis 2-methylcyclohexanol, This is bolded up, right? This is cis. Here's my mirror image. So this is two pairs of enantiomers. This is changed, and then I have the mirror image. So this is two pairs of enantiomers. I actually have 4 here because I don't have any internal plane of symmetry. They don't exist. But let's look at this guy. If I have 1,2-cyclopentanol, if this is bolded out and this is bolded out, I have this internal plane of symmetry where this OH and this OH are reflected. And that gives me meso, because this is actually identical to this one because of that internal plane of symmetry. Notice this doesn't, because this is back and this is forward. So this set has no plane of symmetry. So this is actually a pair of enantiomers. So to have meso, notice I want to have symmetry. I want to have 2 of the same group that has the potential to reflect internally. Here's another example. If I have cis 4-methylcyclohexanol, I actually only have 2 stereoisomers, because if I cut those atoms right down the middle, it doesn't matter if one is cis and one is trans. The mirror images of this one is equal to that one. This is also equal to its mirror image. So all I have is the cis and the trans. They're both achiral. They're geometric isomers, really, because I have that cis and that trans. When I look at 3-methylcyclohexanol, right? If I want to look at that plane of symmetry, this is different from this, so that's not going to exist. This is bolded out, and this is behind, so that's not going to exist. So what I have is a pair of enantiomers and a pair of enantiomers. Anything in between is going to be diastereomers. In this example, again, if I cut this in half, this is different from this. I'm going to get a pair of enantiomers and a pair of enantiomers. And again, these are going to be diastereomeric relationships. So I get 4 stereoisomers, which are 2 pairs of enantiomers. Look, this is the same as this. In one isomer, I have that plane of symmetry. This is reflecting this through this plane of symmetry. So this is going to be meso. There is no plane of symmetry here because this is bolded front and this is hashed backwards. So this is a pair of enantiomers. This particular compound has a maximum of 3 isomers, stereoisomers. I get the meso, which is achiral, and then I get the chiral enantiomers. One more topic is called optical activity. Way in the beginning, we talked about optical activity. So let's look at what this is. Ordinary light has light waves vibrating in all planes perpendicular to its direction of propagation. Plain polarized light has light waves that vibrate only in parallel planes. Polarized sunglasses are an example of this. And you can tell when you put on a pair of polarized sunglasses, you reduce glare, you maximize colors. You're actually affecting the light that's going into your eyes. we can measure this property of optics of light using a polarimeter. And this is looking at the ability of a compound to rotate plane polarized light. If a compound can rotate plane polarized light, it's considered to be optically active. So my individual enantiomers are each optically active. My diastereomers, my individual diastereomers, can be optically active. So these optical properties that they exhibit, meaning they can actually rotate a plane of polarized light, that is what we refer to as optical isomers. If I look at a polarimeter, if I have something that is achiral, the light, I am polarizing to get just one direction of light. But from start to finish, it is not changed. So this happens when I have an achiral compound in my sample tube. I have no rotation here. It is not changing the plane of polarized light. So this is optically inactive. But look at what happens if I put a chiral compound in this chamber. Now notice my plane started out up here and ended up this way. So this is an angle of rotation. This is an alpha. This is going to be an angle of rotation of the light. So the plane is rotated through this angle alpha. This can be measured in degrees, and it's called the observed rotation. This indicates that compound is optically active. If my compound rotates the light clockwise, it is indicated by a plus sign. If my light is rotated counterclockwise, it's indicated by a negative sign. This specific rotation is the actual observed rotation of that optically active substance in a very controlled environment. So every sample has a concentration of 1 gram per mil, and my sample tube is the same length. These two are non-superimposable mirror images. And notice, this is going clockwise, this is going counterclockwise. This is what I call it. I call this an alpha D, which is my specific rotation. My plus is rotating that light in my clockwise direction by 13.52 degrees. My levorotatory compound is rotating that plane of light in the counterclockwise direction. But notice, I have equal in magnitude rotation for these two enantiomers. The direction of rotation is opposite. So when I think of an enantiomer, I have equal magnitude of rotation, but the direction of rotation is opposite. This is really important if I have compounds that are enantiomers versus diastereomers. Enantiomers have identical physical properties. They cannot be separated using regular common physical techniques like distillation. Diastereomers and constitutional isomers have different physical properties, so they can be separated. Let's look at this. we're looking at 2 enantiomers, and then, so A and B are enantiomeric, B and C are diastereomeric. A and B, identical melting points. A and B, identical optical identical solubility. Notice the optical activity, one is plus, one is minus, but they rotate by 13 degrees. This configuration is the enantiomer has the opposite SS. We're not really going to talk about the C and the L at the moment. Notice C, I have a different set of properties here. My melting point is different. My solubility is different. This one actually is meso, so it doesn't have optical activity because it's achiral. This is a meso achiral compound because I have that internal plane of symmetry. So this is one of these compounds that only has 3 stereocenters. I have the enantiomer, and then I have that meso structure. This is called a racemic mixture or a racemate. This is when I have a 1-to-1 equimolar ratio of 2 enantiomers. Notice in this case, my physical properties will change, but this is the important part. This is not optically active. Oops. And that's because if I go plus 13 and then I add a minus 13, right? This is one enantiomer, this is another enantiomer. They cancel out and I get 0 optical rotation. So any racemic mixture, which is a 1-to-1 mixture of 2 enantiomers, has 0 optical activity, 0 rotation. Now, 2 enantiomers are going to have exactly the same chemical properties, except how they react with other chiral compounds. Many drugs are chiral, like we saw with ibuprofen. They most often will react with a chiral receptor or a chiral enzyme. And again, like we talked about before, that enzyme is geared to bind to just one of those enantiomers because those groups are presented in different spatial orientations. In some cases, one enantiomer can be effective and the active ingredient in a drug, and the mirror image may actually be ineffective at best or toxic at worst. So for example, if I look at S-ibuprofen, or actually if I look at S-naproxen or R-naproxen, right? This is naproxen. This is the Aleve. This is the other pain reliever out there. This is actually the anti-inflammatory agent. This is a liver toxin. The only thing I have is a different configuration on that chiral center. So sometimes I have to separate isomers before I can market them as drugs, because otherwise I get bad results in terms of my biological properties. When we look at the chirality of biomolecules, most molecules in living systems are going to be chiral. And this is true for both plants and animals. And even though if I have multiple chiral centers, the possibility of numerous stereoisomers exists. Invariably, only one stereoisomer is found in nature. Nature is very stereospecific. Now, instances do occur where I have more than one stereoisomer, is where more than one is found, but they rarely exist together in the same biological system. So this is how, again, enzymes will distinguish between a molecule and its enantiomer. I'm going to have pockets that are tailor-made for fitting one enantiomer into my active site on my enzyme surface. I will not have compatibility if I change that. So this enantiomer does not fit the same bonding site because the way it's oriented, this guy cannot fit here, and this guy is up in a different direction as well. So even if I maintain those pockets, because I have a different enantiomer, I have a different physical spatial orientation, which means this cannot bind to this enantiomer. It can only bind to this one. Enzymes, which most people in this class have are familiar with because they've taken biology. Enzymes have many stereo centers because they're made of amino acids, and amino acids, we have 20 known amino acids, or 20 natural amino acids. 19 of those are optically active because 19 of them have chiral centers, except for glycine. So chymotrypsin has 251 stereo centers. So I have 2,000, or 2 to the 251. That's 2 to the 251. This didn't come out right. Possible stereoisomers. Only one is produced and used by a given organism. Because these enzymes are chiral, most either produce or react with only substances that match their stereochemical requirements. Again, they are stereo-specific interactions. Because these interactions between molecules in biology take place in a chiral environment, a molecule and its enantiomer, or a molecule in one of its diastereomers will elicit different physiological responses. For example, S-ibuprofen is active as a pain and fever reliever. The R enantiomer is inactive. For naproxen sodium, the S is active, and the R is actually a liver toxin. That binds to a different receptor and causes an adverse event. So how do I get two enantiomers apart if I can only use one, like for producing a drug? Well, if I make the racemic mixture in the lab, which is that equimolar mixture of two enantiomers, I can actually resolve them, which is separation of this racemic mixture into enantiomers. In order to resolve, I'm going to put my achiral pair of enantiomers into a chiral environment. This is a chiral enzyme. It will only react with one of those isomers. So this is a hydrolase. It actually turns that ester into a carboxylic acid. This is the reaction. This is what comes out of that enzymatic reaction. This is a different molecule. This stays the same. And now I can just get rid of this using physical means, and I keep this guy, and I use that in my drug format. This is the end of chapter 6, and later on we will continue the course with chapter 2, looking at acids and bases in organic chemistry. Hopefully this helped. Please come to the Zoom sessions for online or for active problem solving help. Usually people need to practice this a lot in order to get it down. And it's really hard to explain without my models showing everything in 3 dimensions. So hopefully you have those models and you're taking it to heart that you should be building things and looking at the models so that you can actually start looking at these 3-dimensional arrays, especially when they're different, like when I have a set of enantiomers or a set of diastereomers. All right, good night, everyone.